Okay, in a previous video, I demonstrated how light is transferred in the photosystem 2 to photosystem 1 to then NADPH. This is an overview, a much wider picture of what's actually happening. So this is still the same thing that we were talking about previously, but again, just a wider picture. Here you have your photosystem 2, okay, and we know what happens inside of that. And here you have your plasticoinine protein with your cytochrome complex protein, and then you have your photosystem 1, your ferredoxin, okay, and then your NADP reductase. All right. So these individual molecules we've already talked about in detail. So we are just going to give an overview of what is happening once again in this system. Okay. So let me wipe the board clean here. So again, take note of the fact that the beginning process begins in photosystem 2. Okay, you have two things happening in photosystem 2. Number one, you have your light, the energy being absorbed by photosystem 2, and at the same time as your light, you also have your water molecule. Okay, that water molecule gives up its electrons, and then it is split into hydrogen ions and oxygen ions. Now remember, that oxygen atom binds with another loose oxygen atom to make O2. Okay, so those electrons are excited by the light, and then they're passed along the ETC, electron transport chain. And as they're passed along the ETC, at this point here, let me highlight this cytochrome complex. We didn't talk a lot about it in the previous video, but we will in this one, okay? You'll notice that as the electron is being passed the ETC, what is happening is that this cytochrome complex it now has the energy to pump hydrogen ions into the thylakoid space. Okay, so this area all in here within the cell is the thylakoid space, and then outside out here is the stroma. Okay, so as the ETC, let me wipe it again clean here so it can look a little better. So photosystem 2 and the cytochrome complex. That cytochrome complex, as that excited electron is passed through, is pumping hydrogen ions inside the thylakoid space. Now, it continues to pump hydrogen ions. In addition, remember, water is broken up into two hydrogen ions and oxygen. So with water adding hydrogen ions and the cytochrome complex adding hydrogen ions, you are gradually getting an increase of hydrogen ions inside the thylakoid space. Okay, now if you remember, that addition of hydrogen ions is producing a concentration gradient. Okay, a concentration gradient. I'm not the best speller, but it's okay. So that concentration gradient is actually powering the ATP synthase, this protein right here. Okay, this molecule is using those hydrogen ions, pumping them out, and as it pumps it out, it's changing ADP into ATP. Okay, so again, the goal or the idea of the cell, of the plant cell, is to use light energy. Okay, it's using light energy. It's repackaging it or reorganizing it, storing its energy inside of these electrons, and then these electrons are being stored in ferredoxin and then being used by this protein, NADP reductase, and storing that energy into NADPH, and also producing more ATP. And then NADPH, that now very unstable molecule, and ATP, which is also a very unstable molecule, it's taken that energy that was at first in the light, and now it's stored in these molecules, and now they're going to go to the Calvin cycle to make glucose. Sorry, I'm having a hard time here with this. They're going to make glucose or sugar. or we've also seen the formula C6H12O6. Okay, or also CN. Oh, I'm sorry. Yeah, so here, I'll just use this. Making your sugar molecule C6H12O6. Hopefully this overview has helped you gain a bigger picture, a bigger perspective on what exactly is happening inside of the thylakoid membrane with the ETC and where that energy is being stored ultimately.